All right, everyone, welcome to part 3 of the video lecture for chapter 3. This should round out chapter 3. We'll be done with the chapter after this video lecture. You'll recall that in the first 2 video lectures for this chapter, we were talking about the various patterns that we see in evolution. We had just finished talking about number 5 at the end of video 2, which was that similarity decreases throughout ontogeny. We then, in context of that, talked about von Bayer's law, which said that features common to members of a higher taxon, like a phylum or an order, for example, often appear earlier in the developmental sequence of an organism than do defining features of lower-level taxa, like orders and families. And we also talked about Ernst Haeckel's comment that, or statement that ontogeny recapitulates phylogeny and sort of the merits of that. The next pattern evolution that we're going to see, so number 6, is what's known as heterochrony. Heterochrony. Hetero means other or different, and chrony refers to time, so different timing or other time. Heterochrony is an evolutionary pattern where the timing of developmental events changes over time. So we're talking about development, either embryological development or development of a juvenile into adult, what have you. Heterochrony typically takes the form of non-reproductive development rate changes relative to the rate at which sexual characters develop. So non-reproductive development, that is referred to as somatic development. Somatic refers to cells of the body that have nothing to do with reproduction. So somatic development is development of parts of the body that are not involved in reproduction. So everything but the reproductive system, for example. All right, so let's state this again. Heterochrony usually takes the form of somatic development rate changes relative to the rate at which sexual characters develop. This is known as paidomorphosis. Okay, paidomorphosis. Paidomorphosis means child form. All right, child form. And it results in basically a reproductive adult, an adult that's capable of carrying out reproduction that still retains juvenile characteristics. So you get sexually mature adults that in many ways resemble juveniles. All right. Okay, there are 2 ways that this can happen. The first way this can happen is through what's called neoteny. In neoteny, you have reduction in the growth rate of somatic characters relative to sexual characters. So sexual characters and somatic characters, typically develop at specific rates, but in neoteny, somatic characters slow down, or the development of somatic characters slows down, whereas sexual characters continue on at their normal pace. What that means is that an individual becomes sexually mature prior to taking on a fully adult somatic form. And that's one way that paidomorphosis can occur. You end up with a sexually mature adult that resembles a juvenile. And again, that's what paidomorphosis is. All right, the other way that paidomorphosis can occur is through what's called progenesis. In progenesis, you have the reverse situation. In progenesis, you get accelerated development of reproductive organs relative to somatic characters. So somatic characters development doesn't speed up here. It's development of reproductive organs or systems that speeds up. So again, the individual reaches sexual maturity and still resembles a juvenile because reproductive development here outpaced somatic development. And it's important to remember that once an individual reaches sexual maturity, they stop developing. Somatic development stops. So it is where it is once the individual reaches sexual maturity. And in both these cases, neoteny and progenesis, you get a sexually mature adult who has yet to take on a fully adult body form. All right, let's look at some examples of this. Salamanders are well known for this. So what we have on the left is a larval tiger salamander. So these guys are fully aquatic, as larval salamanders typically are, has a laterally flattened tail, uses a paddle for swimming. You see gills here, for example, for absorbing oxygen or gas exchange in water. This guy over here is the adult tiger salamander. It's terrestrial, it's lost the gills, no longer has a laterally flattened tail. It's more adapted to a terrestrial mode of life. Okay, so that's a metamorphic transformation that most salamanders undergo. This is the juvenile form. This is the adult form. Now, bottom left, what you have is what looks like a juvenile salamander. It's not. This is an axolotl. It's the adult. It's a sexually mature adult, the adult axolotl. But you see it's retained the gills, it's retained the laterally flattened tail. It looks, by and large, just like a juvenile tiger salamander. But this is a result of neoteny, okay? Neoteny, where you had a reduction in the growth rate of somatic characters, whereas compared to sexual characters. So development of somatic characters, like, body tissues and that sort of thing, slow down and therefore lag behind development of reproductive characters. So that's as far as these guys go. They're fully developed sexual adults, fully developed in terms of sexual processes, that is, not in terms of somatic development. But that's where they stop. They don't. Their somatic development does not continue beyond that point. All right, here we have another example of what's called progenesis in salamanders. So in this case, what we're looking at is skull structure. So just as in humans and other mammals, for example, and indeed lots of other vertebrates, the skull of juvenile forms often looks quite different than the adult. Here in the salamander, in the paedomorphic individuals of the genus Thoreus, you have this large gap here between the frontal and parietal bones in the skull. That's a juvenile feature, but it's found in the adult. Also true, these bones that are shown here in blue, these are highly reduced in paedomorphic species compared to what they are in non-paedomorphic species. You can also see that gap that we saw there in the paedomorphic species isn't present in the adults of the non-paedomorphic individuals. So, and there are lots of other features about the anatomy of these paedomorphic salamanders that conform to those same basic structures. All right. Number 7. Oops, sorry about that. iPad. Number 7, or our 7th pattern of evolution, is what's known as allometry. Allometry refers to the rate at which various parts of the body grow during development. Allometric growth, as it's known, occurs when different body parts or regions of the body grow at different rates throughout the developmental period. So, a great example of this is human babies. You look at a human baby, if you think about it, the head seems disproportionately large compared to the body, much more so than it is in toddlers or adults, certainly. So if you take a human baby and scale the body length to match that of an adult human, you can see just how much larger the head is, relatively speaking. We've all seen, kids, 5, 6 years old that seem to have a really huge head compared to their body. And you ask yourself, is that kid ever going to grow into his head? That's an example of allometry, but it can result in dramatic changes in overall body form through development. You look at the skull of a human fetus and compare that to the skull of a chimpanzee fetus, about the same stage of development. They look very similar. But the chimpanzee's skull, particularly the frontal bones of the face and the jaw, grow much more rapidly during development into an adult than do the bones of the brain case itself. Whereas in humans, it's the bones of the brain case that grow more rapidly than the bones of the frontal bones of the face and the jaw in particular. And so you end up with skulls that look very different as adults, even though they start from very similar points as fetuses. This is also true, for example, in the very elongate finger bones of a bat's wing. Those are the result of an increased rate of finger growth during development. And that just gives you a good idea of how dramatically shifts in the rate of growth of various parts of the body can change the overall form of the body over development or through development. All right, so these evolutionary changes then that have resulted over the evolution of these organisms, they result in altered growth rates of various body regions and can result obviously in significant phenotypic change in overall physical appearance. All right, number 8, pattern number 8, we have increases or decreases in complexity. Increases or decreases in complexity. So evolution is often sort of thought of or portrayed as a process that results in increase in complexity through time. And we certainly see lots of examples of that. But the opposite is also true. We also see good examples of where features or structures seem to become simplified over time. They start off from a very complex state and become much more simple over time. And this can be due to a loss of features, a loss of bones, fusing of bones, for example, or various other things. Some good examples of this: loss of eyes in cave-adapted species. I did a lot of work in caves for my doctorate, and you see lots of animals in caves that have lost pigmentation. They're white because there's no light, so other organisms can't see them. So there's no need for pigmentation. They don't have eyes, so because it's perpetually dark and eyes are functionally useless. And so the selective advantage there is that you no longer have to devote energy to developing a complex structure like an eye. You can save that energy in a very food-limited environment for other things. We see reduction of digits in hands and feet, especially in the vertebrates over the course of their evolution from, in some cases, 7 or 8 digits down to 5. Here we see the reduction in the number of bones in the lower jaw of mammals through time. There's a very clear and very good fossil record going back to species like Dimetrodon, which is an early mammal-like reptile, lived maybe 260 some million years ago, had a number of different bones. You see 7 different bones that made up the lower jaw. As we go forward in time into their descendants, we see that the number of bones decreases until you get to modern mammals, which just have one, the dentary. All of these other bones have been lost. So that's a really good example of simplification over time. We see the same basic thing in terms of bones of the skull, loss of skull bones. Here we have Eusthenopteron, which is an early lobe-finned fish, an early ancestor of terrestrial vertebrates. Very complex skull structure, all these different bones. It's actually very similar to what you see in modern bony fish. Their skull is just crazy in terms of complexity. Here we have an early amniote. So this would have been a fully terrestrial early vertebrate, basically. And you can see that, or terrestrial vertebrate, you can see that there's fewer bones, still complex, but fewer bones than we had in Eusthenopteron. And then compare that to a dog skull, a modern dog skull, and you see quite a bit fewer bones even so. So this is yet another trend that we see here is, in terms of decreasing complexity. All right, pattern number 9. Evolution often produces trends over time. Trends over time. So we have evolutionary trends then. Trends are repeated changes in a particular direction within a lineage, or sometimes within multiple lineages independently. So repeated, sort of, and to a certain extent, predictable patterns of changes over time. Here's a good example. This is a trend for the shifts in columbine pollinators from bees right here. So here we have bee pollination, or plants that require bee pollination, okay? And to hummingbirds. So these particular species of columbines are pollinated by hummingbirds, and then these on the bottom are pollinated by moths. So you look back at the evolutionary tree, the phylogeny here, and we see that bee pollination was the ancestral condition, okay? And then eventually we have, so right here, the development of hummingbird pollination. We can see that trend moving on this way through these descendant species. And then right here we have the development of moth pollination. It turns out it happened once here, a second time here, and a third time there. So this is a clear trend, right? Moth pollination is the more derived condition, whereas bee pollination is the more ancestral, and hummingbird pollination was sort of in between the two in terms of timing. But this is a clear shift in this direction from bee to moth pollination, with hummingbird pollination being there in the middle. All right. Oops, sorry. Convergence. Yes, I forgot to mention that. So these are examples of convergence, the ones I pointed out, where you had moth pollination evolving once here, and once here, for example. We also see it again right there. I missed that one. Sorry about that. Okay. Our 10th pattern, or evolutionary pattern, is what's called evolutionary or adaptive radiation. All right, evolutionary or adaptive radiation. Radiations occur as species adapt to new or changing environments. A lineage or lineages may diverge into numerous closely related lineages over relatively short time periods. So adaptive radiation is basically what causes the branching pattern that we see in phylogenies, right? Evolutionary trees. So this sort of branching pattern that describes the relationship between ancestor and descendant. That's adaptive radiation. Typically, you'll get one species that will split up into separate populations that then evolve into new species on their own, and so on down the road. And this is basically what's going on in terms of lineages adapting to very different ways of life, moving to new areas, migration to new areas, experiencing new environmental conditions, new pressures, selection pressures, and adapting to those conditions, and in the process of doing that, evolving into new species. And the changes among the various lineages are usually not in one particular direction, as we'll see going throughout the course. Great example of this is Darwin's finches. So we know that there was an ancestral finch from the mainland of South America that was probably blown out to the islands by a hurricane or something like that as they formed and sort of set up shop. And there are 14 different islands, and there are main islands, and there are roughly 14 species of finch. So the idea is that that population that was blown out from the South American mainland sort of landed on these different islands, which had different conditions, different vegetation types, different, somewhat different climatic conditions, different food availability, just different environments. And so each population had to adapt to its own local conditions on the island it was on. And so over time, they've evolved into all these different species. That's what Darwin hypothesized happened to the finches way back in the 1840s. He was saying this. And turns out today, genetic studies have shown that to be the case. All of these modern species of Galapagos finch that we see here, genetic studies have all relayed them back to an ancestral species of finch on the mainland of South America. So the DNA shows us that. So this is just another common pattern, adaptive radiation. All right. The 11th, and I think last pattern, is that of genome size. Okay, genome size. If you think about it, if you think of DNA as sort of being like the hardwiring that dictates how organisms end up, what they are in sum total, to a certain extent, it's reasonable to assume that genome size, how much DNA there is, how many genes there are in the genome, should increase with increasing organismal complexity. You need more instructions to make a more complex organism. That would seem to be a reasonable assumption. There's some places where we see that, but there's actually not a really strong relationship, it turns out, between how many genes an organism has and how complex it is. And that apparent paradox is what's called the C-value paradox, right? The C-value paradox. This is a lack of expected positive correlation between phenotypic complexity and genome size, particularly in eukaryotes. Okay, this is most apparent in the eukaryotic organisms. So animals, plants, fungi, protists, that sort of thing. So if we take a look here at this figure, we can see genome size in picograms of DNA on the x-axis, and we see on the y-axis we have organisms from less complex at the top to more complex at the bottom. This is phenotypic complexity. So first of all, what you see is that if you compare the prokaryotes, which are shown here, to the eukaryotes, prokaryotes do seem to be a little bit less, or have less or fewer genes, less DNA, right? And that makes sense. Overall, the average amount of DNA you would see in eukaryotes is higher than what you would see in prokaryotes. But when you look within eukaryotes, there does not seem to be a clear pattern. Look at birds, mammals, reptiles, fish, and amphibians, right? The vertebrate animals, in some cases, they have less DNA than what you see in algae or protists. So this is the C-value paradox, right? Basically what I'm highlighting right there. That's what C-value paradox is referring to. The lack of apparent positive association or correlation between the phenotypic complexity of an organism and the amount of DNA that it has. We do seem to see that pattern between prokaryotes and eukaryotes, as I've said, right? But we don't see it generally within the eukaryotes. All right, turns out that even though DNA amount varies considerably among the eukaryotes, as you're seeing right here, and not in expected patterns, much of that DNA is actually non-coded repeated sequences, what has often sort of incorrectly been referred to as junk DNA. The transition from prokaryote to eukaryote does correspond to an increase in number of genes, but gene number varies little among multicellular eukaryotes, with a few exceptions. The single-celled eukaryotes do seem to show lower gene number than multicellular species do. So if we look at this figure here, we can sort of see this. So we have number of genes here along the x-axis in the thousands, right? And let me just tell you what these colors are referring to, the color of the bars. So the red are the single-celled organisms, okay, single-celled organisms. So these are the eukaryotes, all eukaryotes. The green is multicellular organisms without tissue organization, so things like slime molds and fungi, for example. And then in the blue, we have plants and animals. So there does seem to be a little bit of an expected relationship where plants and animals, do seem to have larger numbers of genes than certainly slime molds and fungi and single-cell eukaryotes, that sort of thing. But look within the plants and animals. So humans, for example, right down here, maybe 24,000 genes is what the current estimates are. Compare that to rice with 60,000 genes. What the heck is up with that? This is what we mean by this sort of C-value paradox, right? It doesn't seem to make a lot of sense. It seems to be paradoxical that, a rice plant would have 3 times potentially as many genes as a human, for example. But this could well be a result of, genes evolving during the course of animal evolution and then being turned off and sort of becoming vestigial. This is why we have these repeated sequences. Lots of weird things can apparently have happened over the course of genome evolution to sort of wipe away the distinctions or the boundaries between relatively simple organisms in terms of phenotype and more complex organisms. So you don't see that obvious pattern that you would expect to see necessarily. That's the C-value paradox. Okay, so that wraps up the last video lecture for Chapter 3.